Hello students, welcome to my YouTube channel Sharma's Biosciences. Today we will learn about ornithine cycle and in this topic we will cover its mechanism, regulation and significance. So starting from the topic, ornithine cycle, it is also called as urea cycle, and it is basically a cyclic process. It is basically a cyclic process of urea formation which occur in mitochondria of liver cells. It was first studied by Krebs and Hensley, and the organism where ornithine cycle or urocycle is observed, they are all called as ureotelic organisms. It means ornithine cycle or urea cycle is observed in all animals having urea as their nitrogen based excretory product. In this cycle, urea is formed from ammonia. It is formed from ammonia plus carbon dioxide, and they combine to form this is NH2 along with water. So this is urea, and the raw material or ingredient for this urea, these are ammonia and carbon dioxide. So first of all, we will study about the source of ammonia and carbon dioxide, which act as ingredient for the formation of urea within liver cells. So for this we have to cover in brief about the catabolism of amino acids, as the ammonia is derived from metabolism of amino acids and proteins. Similarly, carbon dioxide is derived from respiration as well as decarboxylation of amino acids. So metabolism of amino acids and proteins, it is basically a type of catabolism that is of three main types. First one is oxidative deamination. And in oxidative deamination, what happens that amino acids, they are converted into alpha keto acids, and this require an coenzyme FADH2 and FMN, and ultimately in this process amino group of amino acid is released in the form of ammonia, During this phenomena, these keto acids, they enter into Krebs cycle. We are not going to that much detail. And second type of catabolism of amino acid is transamination. It involves removal of amino group of one amino acid, that is its amino group, it is removed, and then subsequently it is added to other alpha keto acid, and subsequently a new amino acid is formed. This process, it convert the donor of amino group containing amino acid into a new keto acid form, and ultimately a new amino acid is synthesized. And this transamination reaction, it is catalyzed by type of enzymes which are called as aminotransferases. These are called as aminotransferases, and they usually require a coenzyme that is called as pyridoxal phosphate. And this pyridoxal phosphate, it is derived from vitamin B6. And the third type of catabolism of amino acid, it is a type of decarboxylation. That involve the removal of carbon dioxide from amino acid, and this removal of carbon dioxide from amino acid, it is catalyzed by an enzyme that is called as decarboxylase enzyme. Suppose this is the structure of an amino acid. It is having carboxyl group and it is having its own amino group. When it is executed by enzyme that is decarboxylase, and this decarboxylase enzyme also require an other coenzyme that is also called as pyridoxal phosphate, and this ultimately remove CO2 from carboxyl group. This is carboxyl group and this is amino group of amino acid. This is amino acid, and when its carbon dioxide is removed from carboxyl group, the resulting structure is like this. It is having amino group there, having side chain there, but carboxyl group, it is without carbon dioxide molecule, and such resulting structures, they are called as amines, and more appropriately these amines are also called as biogenic amines. Now this decarboxylase enzyme, it is found in liver cells that act on these specific amino acids and convert these into amines. But there are also number of microbial organisms, microbes in the intestinal tract which convert these amino acids into some toxic amines. and they are released in intestinal tract. And basically these amines, they are toxic amines. They are toxic amines. For example, toxic in the category of these toxic amines, there is tyramine, there is also other trypamine, there is also other type of toxic amine, cadaverine, and these are derived from amino acid tyrosine. This is derived from trypamine, it is derived from tryptophan, and this cadaverine, it is derived from lysine. Now ultimately these toxic amines, when they are released by these microbial organisms of intestinal tract, they can lead to a condition that is called as putrefaction. It is called as putrefaction in the intestine. Basically it is a kind of rotten state where number of incompletely oxidized products are increased in amount in the intestinal tract. So this is a type of rotten state and it is called as putrefaction. So this is all about the sources of ammonia and carbon dioxide combine which combine to form urea. And in the next lecture or subsequent part of the lecture, I will discuss about the mechanism and regulation of urea cycle, how it is operated within the hepatic cells. In the second part of urea cycle, we will discuss about mechanism of formation of urea from carbon dioxide and ammonia, and this reaction occur inside matrix of mitochondria, and subsequent phases of this cycle occur in cytosol. So basically here we have a raw material that is derived from the metabolism of proteins, and we have site that is mitochondria, and ultimately other site is cytosol, and subsequently urea is formed, and this urea is then released into the circulation, and from circulation it enters into kidney, and through urine it is excreted out, that is excretion. So first, here proteins, they have catabolic products which are observed in the form of ammonia. So first step is ammonia is fixed with bicarbonates or carbon dioxide, and these are derived from deamination of amino acids, and it is derived from respiration as well as decarboxylation of amino acids. Now these ammonia and carbon dioxide molecules, they are first formed inside mitochondrial matrix. They are first formed inside mitochondrial matrix, and here they are first converted into carbamoyl phosphate, and this reaction, it is catalyzed by an enzyme called as CPS1, that is also called as carbamyl phosphate synthase. And this is the first step of formation of urea from ammonia and carbon dioxide, and it utilizes two ATP molecules for formation of this carbamyl phosphate, where two ADP molecules are released and one inorganic phosphate is released, and other phosphate group is added to the carbamyl phosphate. But here this enzyme carbamyl phosphate synthase, it is regulated by another allosteric molecule that is also called as N-acetyl glutamate. N-acetyl glutamate. This N-acetyl glutamate, it acts as cofactor for carbamyl phosphate synthase. This N-acetyl glutamate, it is synthesized from acetyl coenzyme A and glutamic acid. It is synthesized from acetyl coenzyme A and glutamic acid in the presence of an enzyme that is called as N-acetyl glutamate synthase. And this enzyme is regulated, this synthase enzyme, it is regulated by level of arginine. When arginine amino acid, it is having high concentration within the matrix of mitochondria, then it regulates this synthase enzyme, and subsequently acetyl coenzyme A and glutamic acids, they are converted into N-acetyl glutamate, and it subsequently activates CPS1, that is carbamyl phosphate synthase 1, and that fix ammonia and carbon dioxide into carbamyl phosphate at the expense of two ATP molecules. And these arginines are usually considered as the marker of high protein concentrations or conditions within the body system. And when they trigger synthase enzyme, then subsequently carbamyl phosphate synthase, they are activated to fix ammonia into carbamyl phosphate. This is first step of this reaction, and it is a rate limiting step and it is irreversible. First step is rate limiting step and it is irreversible step. And then subsequently this carbamyl phosphate, it is fixed into citrulline within matrix. This is matrix and this is cytosol. But here this citrulline is formed from another type of intermediates that is called as ornithine. This ornithine, it is synthesized in the cytosol, and through ornithine transporter, it is released into matrix, and here it is fixed by an enzyme that is called as ornithine carbamyl transferase. Ornithine carbamyl transferase, and this ultimately leads to its fixing into citrulline. Citrulline further exits mitochondrial matrix and it enters into cytosol through transporter, that is citrulline transporter, and here then it enters into cytosol. So in the cytosol, uh, citrulline in the presence of another ammonia donor, that is aspartate, it is ammonia group donor, it is fixed into this citrulline and it utilizes two ATP molecule and subsequently AMP and pyrophosphate, it is released. Subsequently it is hydrolyzed, this pyrophosphate is hydrolyzed and it release energy that is utilized by an enzyme called as arginosuccinate synthase enzyme, and it convert this citrulline into arginosuccinic acid, and subsequently this arginosuccinic acid it dissociates into arginine and fumaric acid, and this reaction is catalyzed by enzyme called as arginosuccinase. These arginine and fumaric acid, they are subsequently processed as arginine is subsequently hydrolyzed by it is executed by an enzyme that is called as arginase, and hydrolysis results into release of urea and subsequently ornithine is recycled. But on the other hand, arginosuccinase, when dissociate arginosuccinic acid into arginine and fumaric acid, this fumaric acid it is having a carbon skeleton and it is supposed to enter into tricarboxylic acid cycle, and it is further, this fumaric acid it is further converted into malate and subsequently into oxaloacetic acid, and this oxaloacetic acid is further executed by transamination reactions where ammonia group is added and it is converted into aspartate, and this aspartate it acts as a nitrogen or ammonia donor to the citrulline, and here in case of urea which is derived from hydrolysis of arginine, it is having two ammonia group, one is it which is derived from ammonia of mitochondrial matrix and other ammonia that is mitochondrial matrix and other ammonia from cytosolic aspartate. Subsequently this urea is diffused into the bloodstream, and from where it enters into main circulation, and then subsequently reach to the kidneys, where it is excreted with urine. As we all know that ammonia is highly toxic in the blood, as it can damage brain and its functioning, and sometimes it can prove fatal. So it must be removed at earliest, and during this process it is first fixed into carbamide phosphate and then subsequently converted into a less toxic nitrogen containing substance, that is urea, and this urea, it can circulate within body fluids or blood for some time, but ultimately it is also supposed to be removed from the body through urine. So this is all about ornithine cycle or urea cycle, and it is observed in all ureotelic animals. If you like my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.